Hello friends, in this video we are going to discuss about the photorespiration. So as the terminology suggests, photo means light and respiration is the breakdown of the food substance to produce energy. So it is somehow different from our normal respiration we know, which takes place in the body daily. So what is photorespiration? Simply, this is photosynthetic carbon dioxide oxidation cycle sometimes, or it is sometimes called as glycolate pathway. So photophosphorylation is the process of uptake of oxygen and production of carbon dioxide in light by photosynthesizing tissues. So it is the inverse of normal photosynthesis, that in the photosynthesis we are taking in carbon dioxide and we give out oxygen. But photorespiration is the respiration which takes place in light. However, as I told you before, that it has some difference with our normal respiration. Or in the metabolic pathway that consumes oxygen and releases carbon dioxide, generating no ATP and decrease photosynthetic output. It was firstly discovered by Dicker and Trio in 1959 when they observed that the rate of respiration of green leaves is much higher in light than in the dark. The rate of respiration in green leaves is much higher in light than in dark. Photorespiration is an integral and inseparable aspect of photosynthesis in C3 plants. However, C4 plants, they can prevent photorespiration, and even CAM plants, they prevent this. So let's see about the mechanism of photorespiration. The first thing which you should know is that photorespiration takes place in 3 different organelles. It takes place through the chloroplast, then mitochondria, and the peroxisome. 3 different organelles. It is different from our normal respiration which takes place in mitochondria. So in mechanism, we are saying that photorespiration is an integral and inseparable aspect of C3 photosynthesis. C3 photosynthesis. Rubib carboxylase, which is the main enzyme of the dark reaction, also catalyzes another reaction which interferes with the functioning of Calvin cycle. So the another reaction catalyzed by the rubib carboxylase interfere with the normal Calvin cycle reaction. So why does this happen? This happens because the rubib carboxylase active site has the ability to bind both carbon dioxide and oxygen. So if carbon dioxide is fixed in the active site of the rubib carboxylase, then the Calvin cycle will continue. But if oxygen is fixed, then photorespiration will occur. With increase in temperature and oxygen concentration, when the temperature and the oxygen concentration increases, and this increases always in the day, because oxygen is produced in the day by the process of photosynthesis, and the temperature normally rises in the day when there is sun. Rubib carboxylase show greater affinity to oxygen than carbon dioxide. Therefore, in presence of higher concentration of oxygen and high temperature, rubib carboxylase acts as oxygenase and converts rubib, that is ribulose 1,5 biphosphate, means it is ribulose but has 2 phosphates in the first carbon and in the 5th carbon to 3 carbon compound, phosphoglyceric acid PGA, and the 2 carbon compound phosphoglycolate. So what is different here from normal photosynthesis is that there is no fixation of carbon dioxide here. So the 5 carbon compound is broken down into 3 carbon and 2 carbon compound, and the 2 carbon compound is what is called phosphoglycolate. The phosphoglycolate is quickly converted into glycolate, that is glycolic acid. That's why I told you that sometimes this cycle, this reaction is called as glycolate cycle. This reaction takes place in the chloroplast. The glycolate then moves into peroxisomes. Then in the peroxisomes reacts with oxygen and gets oxidized to glyoxylic acid, glyoxylate or glyoxylic acid. So from glycolic acid to glyoxylic acid, the glyoxylic acid is aminated, means the amine group is added. And as we know that amino acids, they have two groups, amino group and the carboxylic group. So if you are taking the carboxylic acid and then you aminate it, you add the amino group, you convert it into amino acid, is aminated and gives rise to amino acid glycine. So from peroxisome, the glycine is transported to mitochondria. Here the two molecules of glycine interact to form one molecule of each serine. So the two molecules of glycine, they interact to form one molecule of serine, carbon dioxide and ammonia. So when the two molecules of glycine, they interact, yes, they form one molecule of serine, carbon dioxide and ammonia. Serine is also an amino acid. Carbon dioxide is then released out from mitochondria and the byproduct of photorespiration. So photorespiration produces carbon dioxide as the normal respiration. However, it does not produce energy. So serine is deaminated to form PGA. Deamination means the removal of the amino group to form PGA, that is phosphoglyceric acid. The latter, that is PGA, passes into chloroplast to enter Calvin cycle. The process of photorespiration, also called the photosynthetic carbon oxidation cycle, involves three organelles, that is chloroplast, peroxisomes and mitochondria. Photorespiration is also called the C3 cycle, C2. It is also called the C2 cycle because of the formation of two carbon intermediate, that is glycolate. So as you can see, if the carbon dioxide is released during this process, means we are losing carbon. This is not again the process of gaining carbon, but the process of losing carbon. So photorespiration does not produce energy at all, or reducing power. Rather, it consumes energy. Further, it does not do the work of photosynthesis and results in the loss of fixed carbon in the form of carbon dioxide. It is estimated that as much as half of the photosynthetically fixed carbon dioxide may be lost by photorespiration. Therefore, photorespiration is a highly wasteful process. Photorespiration generally occur in temperate plants such as beet, bean, cotton, rice, wheat, barley, etc. So tropical grasses such as maize, sugarcane, sorghum, and sardine, etc., they lack this process. And we shall see later that these plants, which are temperate plants which undergo photorespiration, they are called C3 plants, while the tropical grasses like maize and sugarcane, they are called C4 plants, as we shall see in the next lecture. So we have some of the factors here affecting photorespiration. Photorespiration increases with increase in temperature, intensity of light, concentration of oxygen, and age of the leaf. Also, we have some of the significance of photorespiration. It is true that photorespiration causes the loss of about half of the carbon dioxide fixed, but it has some of the significance. The first significance is that it helps in the dispersion of the energy when the stroma gets closed during daytime because of water stress. So if the stomata they close during daytime, then more of the sunlight energy is absorbed and it's not used. If it is used, some of the energy remains there. So without the process of photorespiration, that energy may cause maybe bleaching of the chlorophyll and etc. So dispersion of the energy by the photorespiration process is very important to the plant. And the second thing is that photorespiration protects the plant from photooxidative damage by dispersion of excess excitation energy. So without photorespiration, the plant or the chlorophyll molecule could be distracted by the process of photooxidative damage. And as you can see here, this is our diagram of the mechanism. Lubib accepts oxygen and then it produces glycolate plus PGA. Then the glycolate loses phosphate to produce glycolate. Phosphoglycolate loses phosphate to produce glycolate. Then they move from chloroplast into peroxomes. Glycolate in the peroxome, glycolate react with hydrogen peroxide to form glycine, and then from there, that is here it is aminated. The amino group is added to form glycine. From there, glycine moves into mitochondria. In the mitochondria, glycine is deaminated by the removal of ammonia. Then it is decarboxylated by the removal of carbon dioxide, and then it is oxidized by the removal of oxygen to form serine. Then serine moves again back to peroxome, where it is converted into hydroxyl pyruvate, and the hydroxyl pyruvate, it is reduced by the NAD. It is NAD to form glycerate. This is glycerate, not glycolate again. So glycerate moves into the chloroplast, where it is phosphorylated to form phosphoglyceraldehyde. So here is the Calvin cycle occur here. Here Calvin cycle can occur. This phosphoglyceraldehyde can be used in the Calvin cycle. So this process, in this process of photorespiration, the Calvin cycle occurs, but the Calvin cycle occurs after the loss of many of the energy in the movement of these molecules from one organelle to another, and even the loss of the carbon dioxide, because as you can see here, there is no carbon dioxide fixed. However, the Calvin cycle occurs, but the carbon dioxide is lost. So after every cycle, you may see that the number of carbon dioxide molecules can decrease. Why? Because they are lost in this cycle of photorespiration. So photorespiration has no relation with normal respiration, which occur throughout the day and night in all types of living cells. In order to differentiate from photorespiration, the normal respiration is often called dark respiration. So here you have some of the differences. This table summarizes the difference between the dark respiration and the photorespiration. It occurs inside photosynthetic cell. It occurs in all living cells. Takes place only in the presence of light. This takes place even in light or in dark. Its substrate is rubib. The substrate here is commonly glucose. The substrate is always recent formed. The substrate may be recent formed or stored one. End products are carbon dioxide here. It's not carbon monoxide. End products are carbon dioxide and PGA, but here the end products, they are carbon dioxide and water. Both the end products are utilized in photosynthesis. Here the end products are not recycled. It occurs in chloroplast, peroxome, and mitochondria. Here it occurs in cytoplasm and mitochondria only. The toxic hydrogen peroxide is produced during the process, but here hydrogen peroxide is not produced. It is wasteful process and does not produce energy. It produces energy for cellular maintenance, synthesis, and work. It increases with increase in concentration of oxygen. It is not influenced by the change in the concentration of oxygen. So that is all about the photorespiration. And in the next lecture we shall go to see on the C3 and C4 plants, as well as the glands anatomy of the C4 plants, before discussing about the Hatch-Slack pathway. So see you later. Subscribe to the channel for more updates, and just share the channel to your fellow students so that they can get this knowledge. For any question or concern, just comment below. Thank you.